Robotics. We are working our way through our new theme on challenges or open problems in the field. So in this module, I don't have any answers for you. I only have problems and partial solutions to those problems. So just keep that in mind. We started last time with modularity. As we've seen in many experiments now, we have one robot, one fitness function, one task, one simulated or real environment. How do we scale up to many robots, many environments, more complex fitness functions? Whatever that solution is eventually going to be, it's going to involve modularity in some way. Why is modularity necessary to scale things up? Regardless of whether we're talking about robotics or engineered systems, biological systems, modularity is everywhere. How does it help scale things up? It helps keep things organized. In what way? Maybe a little bit more detailed.
If you want to change something, you don't have to change in multiple places. You can just change in one place and it will affect the whole school.
If you modify one module, it does not affect the other modules if you have a modular system, right? Imagine trying to build an ever more complicated machine where if you make any one change, it will disrupt function everywhere, right? Makes things very difficult to design and complicate function over time. Okay, we're going to look at two evolutionary algorithms today which have been applied to robotics, neat and hyperneat. So these are solutions to a problem which we will jump into now. What is the problem that neat and hyperneat were designed to solve? So this is a problem that comes from neural networks known as the competing conventions problem. What is the competing conventions problem? This is a problem that arises when you introduce sexual recombination into your evolutionary algorithm. So in all the evolutionary algorithms that you're implementing in assignments 6, 7, 8, and 9, you're only going to be using mutation. You are not going to be crossing two genomes and mixing and matching genetic material from two genomes. The reason why is it's very difficult to get that to work well. Mother Nature discovered sex very early on, and there is a good reason to genetically combine material from two or more parents, but it is a very difficult thing to get right. We're going to look at that in the context of neural networks. So I have two cartoon neural networks here. Each has two input neurons, one output neuron, and three hidden neurons. Let's imagine that in these two cases, the three hidden neurons are computing subfunctions, A, B, and C, and those subfunctions need to be combined in some way to do whatever it is we want this neural network to do. So once again, if you'll cast your mind back to our earlier discussion about neural networks, and we talked about the neural network that solves the XOR problem, there were two hidden neurons in that neural network. One hidden neuron computed the AND of the inputs, the other hidden neuron computed the OR of the inputs, and then the AND and OR partial results were combined in a certain way to compute XOR, right? So to get a neural network to compute XOR, you needed to compute two partial solutions and then combine those partial solutions into the overall solution. In this cartoon example here, imagine there are three subfunctions, A, B, and C. Imagine we are evolving our neural networks. So we have a population of neural networks. We have these two here, which are doing pretty well. Let's imagine that all two networks, whatever the synaptic weights are, they're producing almost A, almost B, and almost C, but you'll notice that they have competing conventions. In one of them, in the first one, the first hidden neuron computes A, and in the second one, it's the third hidden neuron that's computing A. Right? Not yet a problem. You could imagine two neural networks that are evolving weights to perform the XOR function. And in one network, the left hidden neuron does AND and the right hidden neuron does OR. In the second neural network, it's the other way around. The first hidden neuron does OR and the other one does AND, right? Okay. Let's further imagine that this one here is doing a really good job at A and B, but not so good at C. The other one is doing pretty well at C, but not very well at A and B. You can probably see now where this is going. We have two neural networks in the population, and those two neural networks have modules inside of them, and those modules are solving different parts of the problem, but no one neural network has the whole problem yet. So we would like to try and cut these two networks in half, or cut them in a certain way that we could pull out the good C and the good A and B from the two parents and glue them together in a child. So we end up with a child that is good at A, B, and C, right? That is why Mother Nature discovered sex early on. If you can do that, you can bring together the best of both worlds in parents that are contributing genetic material into the same offspring. So far, so good? Okay. I want you to mentally take a straight line and cut these two neural networks with a straight line that you can combine A, B, and C in a child network. Hopefully, if I've set this up right, there is no such straight line, right? So although we have two partial solutions that, if combined, would produce a child that's better than either parent, there is no cut point. There is no place, at least with a straight line, that you could imagine cutting this. There's probably a tortuous path that you could trace with scissors through both networks that would do the job. But this is sort of meant to be a cartoon example to illustrate this problem. So to get crossover right, we need to be able to somehow bring together this the good material, even if there's competing conventions, even if different subproblems are being solved at different parts in the neural network. Question?
This process is also complicated by the fact that the weights are equally, if not more important than the nodes, in the sense that the node is just a conduit for the values passed through the weights. So we'd actually want moving whole weights around, not just the node C.
Correct. You're absolutely right. So I'm using sort of some lazy terminology here when I say copy A. What I mean is the red weights that are going into A and the red weight that is going out of A. You're absolutely right. We want to really combine subsets of synaptic weights, right? But how do we know that the red weights, which are computing A in this network, are the same as the red weights over here? They're obviously not actually red in the evolving population. It's difficult to identify similar structure in neural networks, and as we'll see later, in robots altogether. So a number of years back, Stanley introduced an algorithm which we're going to talk about now called NEAT, and it is indeed a NEAT algorithm. NEAT stands for neuroevolution, so we're evolving neural networks, neuroevolution of augmenting topologies. So topologies is a reminder that we're in the NEAT algorithm, we're not just going to be evolving weights, we're going to be evolving the topology or the architecture or the structure of the neural network itself. This is something we mentioned back towards the beginning of the course. It's relatively easy to evolve synaptic weights for networks with fixed topology or fixed structure. But evolving populations of neural networks in which the architecture itself, the number of neurons and synapses and the way they're all wired up, for those to grow over time, over evolutionary time, is a difficult thing to do. And NEAT is probably the best algorithm still out there for doing that 19 years in. It's a pretty standard algorithm these days. Remember that an augmenting means that the topology itself might grow or shrink or change over evolutionary time, the NEAT algorithm. Okay, as promised, NEAT was designed to help solve the competing conventions problem. So let me tell you about the intuition behind the NEAT algorithm, and then we'll dive down into its details. In order to solve the competing conventions problem, you've got to be able to identify, or the evolutionary algorithm has to be able to identify similar substructures, like the sets of red weights in both networks, even though those structures might be at different places in different networks in the evolving population, right? A difficult thing to do. The intuition that we're going to use here is to exploit the fact that these are networks that are being evolved. So in this cartoon example, we have two neural networks in evolving population. But these networks in turn probably had parent networks, which in turn had parent networks. And it may be, in many cases, it might be that if these two neural networks are surviving in the population, they might have actually had a common ancestor. There may be one neural network which is similar to both that led to these two offspring, right? So generally speaking, if you cut open a human body, you find the same organs in more or less the same place, but not quite, right? There are certain differences. And although I'm not a doctor, I don't know if I could recognize certain organs. I know that the stomach in one human and the stomach in another human are the result of an ancestral stomach from a common ancestor in our lineage way, way back. Right? So same thing actually with computers. If you open a Mac or a PC, the CPU is going to be at different positions on the motherboard, but those two CPUs in those two laptops are the great-great-great-great-great-grandchildren of an ancestral CPU back in the very early days of personal computers. So those two CPUs have different positions in the motherboard. They have different functions. They act slightly different in a Mac and a PC, but they generally do the same thing because they are the result of an idea that was built once in one of the first laptops, the very early days of computing. So we are going to give the evolutionary algorithm a way to track different or tag different structures inside these evolving neural networks. And that tag is going to be the same in multiple networks. So here you can think of the red as the tag. Different parts of the network that have the same tag, that tag points back to a common ancestor. That tag is indicating that those two parts of the network came from the same single part in a single ancestral network. The idea then is if we find common tags, when we cut our neural networks and combine structures, we want to make sure that we do not put structures, we do not copy into the child parts that have the same ancestral tag. So in this case here, if we were to naively cut, if we were naively to cut between B and C, if we were to cut between the second and third hidden neuron here, and also cut between the second and third hidden neuron here. In the first child, we'd copy in the red and the blue weights, giving us AB. So we'd copy everything on the left side of the cut in neural network 1, and we'd copy into the child everything on the right side of the cut in child 2, which would be A. And unbeknownst to us, we would end up with redundant structure in the child that has two copies of A and does not have a copy of C. So this child is likely to be worse than either parent, right? Same thing if we then keep the same cut point and we copy in everything from the left side of the cut point in parent 2. So C and B go into child 2. We take the same cut point and we copy everything on the right side of the cut point from parent 1, which is again another copy of C. So we're going to, in the need algorithm, we're going to introduce these tags, and when we're cutting, we're going to make sure we don't get two copies of structure that has the same ancestral tag. So far, so good? Intuition makes sense? Okay. So how do they do this? They're going to encode in neat a genome. Remember, the genome or the genotype is the blueprint of the thing, and the thing is the phenotype. The genome is going to be two vectors, two lists, actually, if you like. The first list contains information about the neurons in the neural network, and the second list contains information about the connections or the weights. So here's one genome, and here's the resulting phenotype, which is a neural network. The first list indicates that we have 1, 2, 3 sensor neurons and one hidden neuron and one output neuron. And we're assigning an ID number to each of those 5 neurons. Pretty straightforward. Then we have a list with 6 elements down here, and each element in the synapse list has information about a particular synapse. So the first synapse connects neuron 1 to neuron 4. That's this synapse here. Here's its particular weight. We're going to include a binary tag, which allows evolution, if evolution flips that switch, to make that synapse present or absent. So an additional detail. And then we have an innovation number. So this is the ancestral tag here. And in this cartoon example here, we have a number of innovation tags, which we'll talk about in a moment. So this produces a particular neural network. You'll see there's a recurrent connection here because we have one synapse that is connecting neuron 4 to 5 and a second synapse that is connecting neuron 5 back to neuron 4. Okay. We have mutation in the NEAT algorithm. The mutation can add or remove an element to either list, which will result in adding or removing a neuron or a synapse. And mutations can also modify values inside the element of a list. So you can imagine a mutation that flips a disabled synapse. So in this particular genotype at the moment, there's a synapse that connects neuron 2 to neuron 5, but it is disabled at the moment in the genotype. A mutation that flips disabled to enabled would add that synapse to the neural network. The NEAT algorithm introduces a number of new biological mechanisms into our evolutionary algorithm that we haven't seen yet. One of them is this ancestral tagging method. The other one is the introduction of junk DNA. This is an interesting twist in the history of evolutionary biology. There are many, many parts of your genome that seem to be inactive over your entire lifetime. And there's a copy of that quote-unquote junk DNA in every cell of your body. Seems like a lot of dead weight to be carrying around in an organism. There's been a lot of argument and controversy in the evolutionary biology literature about whether junk DNA is actually junk. It's hard to tell and sort of goes back and forth. If you're interested, it's a worthwhile literature to dive into and learn a little bit about. We won't talk about that today. We're going to focus mostly on this ancestral tagging idea. Okay, so that's mutation. As I mentioned, the NEAT algorithm was designed to try and solve the competing convention problem, which arises when we're trying to sexually recombine genetic material from two parents. So here's another slide about mutation. I think we just talked through this, pretty straightforward. Let's have a look at crossover. So we've got two parents in the population, and these two parents have relatively high fitness in the population, so they've been selected to produce a child, the child is going to result down here. You'll notice that both parents actually also have different topology. So this makes things really difficult. How do we cut these two networks in half and have any chance that when we glue the two halves together, we're going to get something that is as good as or better than the parents? This is a very dangerous prospect, right? I cut your Mac and your PC in half and glue the two halves together. Exceedingly unlikely that I'm going to get a new computer that's better than the original two computers. Okay, so how does this work? Remember that although we have these two networks, their genotypes are two lists. In this schematic here, we are only looking at the synapse list, not the neuron list. We're not going to worry about that for the moment. So parent 1 has 5 synapses, but one of them is disabled. So you'll see there's 4 synapses in parent 1. And we have 1, 2, 3, 4, 5, 6, 7. We have 7 synapses in parent 2, 2 of which are disabled. So 1, 2, 3, 4, 5 existing enabled synapses in parent 2. We've written this in short form, so we're not writing the weights. We're just showing the connectivity. And the integers that you see at the top here are the innovation numbers. And if you look carefully, you'll see that some of these integers appear in both parents and some don't. So the first synapse has an innovation number of 1 in both parents. And that innovation tag reminds us that that synapse with a number 1, they are both descendants of some synapse that also had a number 1 back in an ancestor. So I just realized I forgot to mention one additional detail here. Yeah, okay. I forgot to mention one additional detail here is during mutation, as we mentioned before, mutations can add, modify, or remove a neuron or a synapse. Whenever we add a connection or a synapse, whenever there's a mutation that adds a synapse, that's a new piece of phenotype, a new gene, a new piece of phenotype. We have a global counter. We have an integer that we are recording throughout the NEAT algorithm. And every time we add something new, we take the new number from that global counter, which in this case is 7, and we assign that value of 7 to that synapse. The next synapse that's added by mutation to any other network in the population is going to get an innovation number of 8, and then 9 and 10, and so on. So at this point in time, in all the neural networks from the current evolutionary run of NEAT, no synapse has an innovation number of 7 except this new synapse that we just added. As evolution continues, and if this neural network survives and produces offspring into the next generation, those offsprings which inherit this synapse from this parent, that synapse in that child has an innovation number of 7, right? It's inheriting that. If this parent produces 2 children, and both of those children have this synapse that connects neuron 3 to 4, both of those synapses in both children have the same number, the number 7. So if we see that number appearing in both children, that's a reminder that that piece of neural network was inherited from a common ancestor. So far, so good? Okay. Now, we don't know what function, for example, synapse 1 is performing in both networks, but we can be pretty confident that that synapse is contributing to a similar, if not the same function in both children, right? Your Mac and your PC, the CPUs in your Macs and your PCs do similar things. They don't do completely different things because they share a common functionality from the ancestral CPU back in the early days of computers, right? Your stomach operates slightly different from my stomach, but they do more or less similar function because evolution hasn't had long enough to diverge or differentiate this function. They have similar function to each other and similar function to the ancestral phenotype, the ancestral function. Okay, so we want to make sure then that when these two parents produce one child, they only get one copy of that redundant structure in both parents. So we're going to take the 5 synapses from parent 1 and the 7 synapses from parent 2, and we're going to line up those two lists next to one another, but we are going to line them up sequentially. So we'll start with the first element in this list, the first element in this list. If they have the same innovation number, we line them up on top of one another. We go to the next synapse, which has an innovation number of 2. There's another synapse in this list that also has an innovation number of 2. We line those up. We come to the next number in both lists, the next lowest number, which is 3. There's no 3 over here. So we place 3 in the list, and we leave a gap because there's no corresponding 3 in parent 1. We go to the next highest number, which is 4. There's 2 copies of 4 in both parents. We go to the next highest number, 5. There's 5 from parent 1, but none in parent 2, so we leave a gap and we continue. So we now have 2 lists that are lined up with a few gaps, and we have a lined up similar structure, or at least things that have a common ancestor. Right? This is, again, borrowed from how sexual recombination actually happens in nature. DNA material from both parents is actually literally lined up, and bits of that DNA are copied into the children by marching down that list where similar alleles of the same gene are lined up, right? Same idea. Okay, the final step is to produce the offspring genotype from the 2 parental genotypes. Very simple. We just march from left to right, and at every element in these lined up lists, we flip a coin. Heads, we copy in the version of parent 1. Tails, we copy in the version of parent 2. If we get to a gap, we can either, again, flip a coin. Heads we take from parent 1. Tails we take from parent 2. If it's a gap, we quote unquote copy the gap and there's just nothing there, and we continue. Make sense? Question?
Does this mean that every offspring will, at the very least, be as large as the biggest parent?
Not necessarily. So you'll notice there is 1, 2, 3, 4 gaps here. So if, when we're flipping coins and copying, we copy this gap, this gap, this gap, and this gap into the child, there are going to be 4 gaps in the child, and we're only going to get 1, 2, 3, 4 synapses in the child. So the child, if we copied in those 4 gaps, we would get a child with 4 synapses, which is smaller than either parent. So it is not necessarily the case that the child is larger than the parent. Make sense? Okay, so we don't know that we've solved the competing conventions problem here, but even in this cartoon example, if you look at the topology or the structure of the child, it looks similar to the structure of both parents, right? It doesn't look completely different. Now, what a neural network looks like doesn't necessarily tell you its semantics or what it does or what it computes, but it's often a pretty good proxy for that. Make sense? So again, this algorithm was published many years ago, but is a standard tool in evolutionary algorithms and evolutionary robotics for exactly this reason. It allows mutation to make local changes to structure. And if you have a modular structure, that's good news because a mutation can alter part of a neural network without disrupting other parts of the neural network. But we also get recombination, which is a bigger change, right? We're grabbing genetic material from one parent and genetic material from another parent and putting it together. That's a much bigger change than changing one weight of one synapse. But the NEAT algorithm increases the probability that that big change in some cases produces a child that is better than either parent. If we get lucky, we're combining the best of both worlds, like we looked at in the cartoon example here. Yeah? Question?
Does this approach always work?
Does it always work? So at the level of pairs of networks, absolutely not. It is still a low probability event that you're going to produce a child that's as good or better than the parents, but that probability is higher. And since we're looking usually at populations of hundreds or thousands of neural networks, and there are hundreds or thousands of these recombination events going on in any one generation, as long as it's happening some of the time, that helps. Yep.
Do you know of any work that followed this that tried to introduce the idea of dominant and recessive genotypes and stuff like that?
That's a great question. I don't know if anyone has added on the idea of dominant and recessive genes. That would be an interesting thing to look into. You could probably Google NEAT and dominant recessive and find out. Okay. Again, one of the nice things about NEAT also is that it's relatively algorithmically simple. For the grad students or those of you that have made it to assignment 10, you can probably imagine that with relatively few changes to your code, you could switch over to the NEAT algorithm from what you currently have. Yes?
Are you evolving the weights at the same time?
You are also evolving the weights, which I don't think I have a picture here, but there are mutation events that can hit one of the elements in the synapse list and hit the actual weight and change that weight to something else.
Are you doing it simultaneously, or is it you evolve the weights for a number of generations and then you do the recombination?
Yep, that's a great question. Do we do weights for a while and then recombination? Again, this is an old algorithm and there are now hundreds of variants on this algorithm, and that is a very good question. How often should you, for example, mutate weights compared to sexually recombine material from parents? Hard to say. There are approaches out there that introduce adaptive mutation rates, So if you're mutating weights too much and you're not making evolutionary progress, maybe you reduce the rate at which things are mutated. Same thing with the rate at which you recombine genetic material. You can try to, yet again, turn over this task of deciding how often or how rarely to perform different genetic perturbations to evolution. However, that brings up yet another problem, which is how do you adapt the rate at which weights adapt? You get into infinite regress, right? But there's approaches out there that also try and deal with that. Okay, any other questions about NEAT before we push on? No? Okay. Okay, we are now going to talk about HyperNEAT, which, as you can probably guess from the name, is an algorithm that builds on top of NEAT. So as I describe HyperNEAT, just keep in mind that under the hood, we have a population of evolving networks, and those networks are being mutated and genetically recombined into new neural networks to do something. What are those neural networks trying to do? In HyperNEAT, those neural networks are trying to do something specific, which is to paint regular patterns inside a given space. And we're going to see some examples of how this works. These networks have a particular name. They're called CPPNs. So let's just do a little bit of terminology first. NEAT is an evolutionary algorithm for evolving neural networks. HyperNEAT is a way to bias evolutionary search towards particular patterns. We're going to paint patterns inside a space. So HyperNEAT is NEAT that is evolving particular kind of network, and CPPNs are those kinds of networks. Yeah? Okay. CPPNs, let's talk about what they are. They've got it's kind of a mouthful of a word, compositional pattern producing networks. These networks produce patterns. And what do we mean by patterns? Well, let's look at the little cartoon example here. We have a CPPN on the left, and we have the thing that the CPPN produces, the pattern it produces on the right. As you can see in this example here, it's producing a picture of an insect. Let's start with an even simpler picture. So we're going to look at a couple of CPPNs and the patterns that they produce. Let's create a very simple CPPN that has two inputs. We're going to call those inputs X and Y. Why X and Y? Because for the moment we are going to paint patterns inside a two-dimensional space defined by the X and Y coordinates. We're going to visit each point in this two-dimensional space, and every point in this two-dimensional space has a set of coordinates. At the lower left, the coordinates are 0, 0. So we're going to feed the coordinates into the input layer of the neural network. Seems like kind of an odd thing to do to start with, but bear with me. We are then in this very simple CPPN that we're going to start with. We're going to define one output neuron, and I'm going to label this output neuron B for black. So the value that arrives at the output neuron here, we are going to use an activation function to squash this to a value that ranges between 0 and 1 for now. So how much black we are going to place at x equals 0 and y equals 0. So far, so good? Okay. So we have a CPPN with two input neurons, one output neuron, and no synapses. So if I visit this coordinate, feed in these values, how much black are we going to put at this pixel? 0, right? There's nothing arriving down here. We put no black here. We go to x equals 0.1 and y equals 0. How much black do we put here? None. We continue visiting all of the coordinate positions inside this space, and we get a very interesting picture, which is just all white. So we have one CPPN that produces the null image. Yeah? Okay. Let's make things slightly more interesting. Let's add a synapse, and you could imagine that we have a population of these CPPNs that are being evolved by the NEAT algorithm that we just saw. And the NEAT algorithm adds one synapse, and we'll give it a weight of 1. Okay, we feed in 0, 0 to the input layer. What do we get down here? Still 0. We feed in x equals 0.1 and y equals 0. What do we get here? 0.1. So we get light gray. If we continue this along the x-axis from x equals 0 to x equals 1, what do we get along this bottom row? Light gray to darker and darker and darker black, right? We get a horizontal gradient along this row. What happens at the next row? Y equals 0.1. Same thing. We're going to get a gradient that gradually darkens from left to right. So with a relatively simple neural network, there is no horizontal gradient in the network, but the horizontal gradient emerges by allowing this neural network to paint this pattern into this space. Okay, again, hopefully you're wondering to yourself, why would you bother doing this? We'll come back to that in one moment. Let's imagine a slightly different CPPN that has just a single synapse connecting the Y input neuron to the output neuron, and again, a weight of 0. What picture is this CPPN going to paint? A vertical gradient, right? So it's going to start white and gradually darken as Y increases. One of the magic things about CPPNs is if you create a random CPPN, we could throw some hidden neurons in here and wire it up with synapses and recurrent synapses with different positive and negative weights. It will not produce a random pattern. We're not going to get white noise inside the pattern. We are going to get gradients and combinations of gradients and so on. You'll notice if we now come back to the cartoon CPPN we see in the slide here, we have a CPPN that has two input neurons and one output neuron. And this cartoon over here is in color, but just imagine this cartoon is actually in grayscale because we have just one output neuron, which is dictating the amount of ink we put at that position. You'll notice that inside these neurons, there are these inset graphs. What are these inset graphs? What are they visually representing? Remember all the building blocks, the things that make up a neural network. Any ideas? It is definitely a function, right? What is the function that lives inside of a neuron? What's it called? The activation function, right? So up till now, we've talked about activation functions which take the raw weighted sum that's arriving at the neuron and then squashes it in some way, like the sigmoid function. In CPPNs, you can have lots of different kinds of activation functions, some of which don't squash at all. So you'll notice this neuron in here has the identity function, which means that whatever the raw weighted sum is that's arriving at that neuron, that raw weighted sum is exactly what's going out along the outgoing synapses of that neuron. We don't have any sigmoids here, but we notice some Gaussians, which are kind of weird. So the Gaussian, for example, the one sitting here, tells us that if the incoming value is near 0, so the x, you can think of the x-axis in these inset figures as the raw weighted sum. X equals the raw weighted sum. If that raw weighted sum is near x equals 0, then the outgoing value is maximal. It's 1. So the y value, the y-axis here, represents the value that is being sent out along the outgoing synapses. So we have a whole bunch of these different activation functions. And you'll notice, for example, the Gaussian, which is the Gaussian curve, its left side looks exactly the same as its right side. You'll notice in this cartoon example, the left side of this image looks the same as its outgoing. The left side of the image looks the same as the right side of the image. So I mentioned that these networks, CPPNs, are networks that produce patterns. They actually compose patterns together. They are a composition of functions.
What about the heart?
Good question. So what this cartoon is meant to represent are different kinds of regularities that you see in images painted by CPPNs. In the cartoon we did at the board, you could see gradients appearing when we did not squash. We had basically the identity function here, right? If we put a Gaussian in here, and you can again do this with pen and paper, you'll get an image where the left side of the image looks like the right side of the image. You'll notice there's a sinusoidal or trigonometric activation function in this one, which produces, as you can see in the curves, repetition. You can see our little insect here has repeated segments. And then finally, you have the little heart here, which is meant to represent that you can also add in additional details, something that is not bilaterally symmetric that is added here. So CPPNs don't just paint, or they don't just produce patterns, they produce patterns within patterns within patterns within patterns. They compose patterns within one another by composing functions. If you can, for those of you that are mathematically inclined, you can take this cartoon and sit down and write it as a mathematical function. So this is the identity function. So f of x and y is the identity function. G is, I don't even know what that function is. G of x of y is composed inside of h, and h is a function that encapsulates or composes G inside itself. So what a CPPN really is some arbitrary composition of functions, and each function is distorting or warping the coordinate frame. It is changing it in some way, and the composition of coordinate transforms at the end are read out as the amount of black, for example, for the moment. Make sense? So the question about what is the heart doing here, it's just a reminder that we can get patterns within patterns within patterns in a CPPN. Question?
The heart is also supported by.
The heart is also, exactly, so you could actually imagine within that non-symmetric, single appearing structure, there is bilateral symmetry. You can get fractal structures with CPPNs pretty easily. Okay, so we've talked about the how of CPPNs, how they work, but I've deferred the discussion of why CPPNs. If the NEAT algorithm, which is running in the basement of this algorithm, the NEAT algorithm is evolving these neural networks. Remember, each neural network is painting a different picture. So at the moment, the HyperNeat algorithm is searching over the space of possible images, or it's searching over the space of things that paint particular patterns. The CPPNs bias evolution to search for patterns that have regular structure in them, right? Imagine we had a population not of neural networks, but we had a population of matrices, and those matrices actually represented the amount of black at every point in the matrix. If we were evolving genotypes that are matrices, and we started with random matrices, they would be producing random patterns. If instead we start with evolving populations of random CPPNs, they do not produce random patterns. They produce random gradients or random waves of black across the image. What about this CPPN? What pattern does it produce? It's a diagonal gradient, right? So I randomly added a new edge, but we are biasing search towards regular structure. Yeah? So the hyperneat algorithm includes yet another biological mechanism into this evolutionary algorithm, which is the fact that evolution doesn't put random cells together to produce a human being. Evolution itself evolved to produce regular structures, right? And then mutations alter those regular structures, but they don't move one cell around one at a time, right? It's not a scalable solution. And you can imagine that random cellular rearrangements of a successful organism, the vast majority of random cellular rearrangements of an organism is fatal. But moving a stomach from here to here, or adding a few more neurons to a nervous system, or removing a few, or altering how those neurons are put down in the developing embryo, that might have a higher likelihood of success. So this is something that's often missed for those that are new to evolutionary biology. Evolution does tinker. She is a satisficer, but she doesn't tinker randomly. Mutations are random. Recombination events are random, but they produce non-random results. They move things, they alter things in ordered ways. So far, so good? Okay, so let's continue to build up our intuition of CPPNs before we actually put them to work in evolutionary robotics. CPPNs have a lot of cool properties. One of them is that they are resolution agnostic. So what do I mean by that? In our cartoon example here, we had a resolution of 0.1. As I marched from position to position within this two-dimensional space, I was updating by 0.1 every time. And for this CPPN, that produces a horizontal gradient that gets darker and darker as we march up and to the right. What happens if I don't change the CPPN, but I ask the CPPN to paint its pattern again, but now to paint it with this resolution? What do we get?
Better resolution.
Better resolution, but the exact same pattern, right? So this is kind of interesting. We could evolve things. At the moment, we're just evolving pretty pictures. We could evolve things at low resolution, and then later on in evolution or at the end, we can increase the resolution without disrupting the pattern that's produced by the network. Kind of interesting, right? Okay. Another thing that's interesting about CPPNs is they are also agnostic to the dimension. So at the moment, we've been painting pictures in 2 dimensions. Let's imagine we wanted it to paint pictures inside 3 dimensions. What would we need to alter in this setup for CPPNs to paint 3D pictures rather than 2D pictures? Add Z, absolutely. Okay. This CPPN will paint a pattern that marches along the grand diagonal inside a cube from the lower front left to the upper right back.
Wouldn't that be a sphere with that activation?
Sorry, yes, you're right. Let's go back to the identity. Let's go back to the identity activation. You're right, it would be a sphere if we use the Gaussian. Exactly, good point. Which reminds me, in hyperneat, mutations can add and remove neurons or synapses to CPPNs. They can cross genetic material from 2 CPPNs, and they can also mutate the activation function that is inside a CPPN. So again, this CPPN paints a gradient that darkens along the grand diagonal inside a cube. This mutation event that changes the identity function to the Gaussian function is going to change the pattern to a darkening sphere as you approach the center of the XYZ cube. So far, so good? Okay. Let's say instead of painting pretty pictures, I want to get a CPPN to create a movie for me or an animation, a picture that changes over time. What change would I make to the CPPN?
Theoretically do it with 3rd dimension.
Okay.
Shift through
Absolutely, right? So as you've figured out, we need 3 dimensions, x and y for the picture itself, and then a third dimension, which we'll just call t for time, or f for frame in the animation. So now we can set t equals 0 and paint 1 frame in the animation, set t equal to 1, and we'll get a different picture because the actual frame index or the time index is feeding into the neural network, and we get an animation. Right? Kind of interesting. We could evolve CPPNs to produce different kinds of animations. I think you might get the idea here that CPPNs are generative. They generate patterns, and those patterns can be in any spatial and temporal. You can paint patterns across any spatial and temporal dimensions you want. There's an interesting paper where somebody used CPPNs to paint notes onto a music sheet, and now you have a CPPN that's painting music, right? So music unfolds over time. You have the vertical axis, which is frequency of the notes. It's an interesting exercise, again, as you start to build up an intuition for CPPNs. You can get them to generate lots of different kinds of things. Images, animations, music. We are now going to switch and create a CPPN that is going to also paint a pattern in space, but that pattern is not a pretty picture or an animation or a piece of music. It's a neural network controller for a robot. It's kind of meta, right? We have a neural network, which is a CPPN, and it's going to construct another neural network. Seems like kind of a roundabout way to create neural networks for a robot. Why would we do such a thing? Why not just evolve weights for neural networks directly? Why evolve CPPNs that are going to paint weights onto the neural controller of a robot? What might be the potential advantage of doing things in this way? No idea yet? Okay. Idea?
Well, for one, you can that you can have symmetry. So if your robot has bodily symmetry along multiple axes, you can have the weights reflect that.
Absolutely, right? So remember our discussion about locomotion from last week? There are a lot of symmetries in legged gaits and various other ways that animals and machines can efficiently get themselves from point A to point B. If we have a bilaterally symmetric organism or machine like us, it might make sense to paint weights inside the neural network so that the weights themselves are bilaterally symmetric. We actually already saw an experiment where that was done manually. Which experiment was it that we looked at? Where they forced bilateral symmetry in the weights of the neural controller. You remember the hybrid dynamic walker that walks down the hybrid walker that walks on flat ground, right? It had this central pattern generator that was inputting a regular beat, and that beat would arrive in antiphase in 2 identical neural networks, one controlling the left leg and the other one controlling the right leg, right? So in that case, we can imagine bilateral symmetry makes sense. What about a 4-legged animal that also has a gripper on the front and is carrying something so that its weight from time to time shifts over its front legs? Should we have an identical neural controller for each of the 4 legs? Should the 2 front legs have the same controllers and the 2 back legs have different controllers? Whatever it is, it's probably a composition of patterns of weights. But what exactly that composition of repeated weights should be, who knows? That's what we want to see, or what the investigators in this case, Clune and his colleagues, wanted to see in this paper, in which they're going to use HyperNeat to evolve gaits for a quadruped. Okay, so quadruped, we know we have 4 legs. Again, this neural network is drawn a little bit different from what you've seen before. This neural network is going to be drawn in 3 dimensions, not 2 dimensions. So we've got one two-dimensional plane that embeds the input neurons, and we'll talk about them in a moment. We've got 20 input neurons. We have a hidden layer, which is also embedded in a two-dimensional plane. And then finally we have an output layer back here. So let's start with the input layer. We have 4 legs, and each of those legs is going to have a forward and back rotational joint in the hip, and an in and out rotational joint also in the hip. So no knees, no elbows, no ankles, no wrists in this case, just 2 degrees of freedom for each of the 4 legs. So we have I'm sorry, there is a knee here. I take that back. 3 mechanical degrees of freedom for each leg. Forward and back, in and out, and the knee which also rotates. Yeah? Okay. So we've got a total of 12 mechanical degrees of freedom. And we're going to feed into the input layer at every point in time the 12 angles of those 12 joints at that point in time, right? Our proprioceptive sensors. We are also going to feed in the touch information arriving from the 4 feet, which feet are in contact with the ground or not. We are also going to feed in 3 additional sensors, which are the yaw, I always get this wrong, yaw, pitch, and roll of the quadruped's main body. So we're feeding in the orientation of the robot's body over time. And then finally, a sinusoidal pattern that we're just going to feed in to the input layer. What is this sinusoidal neuron called?
Is it for the walking?
It's there because presumably it will help with walking.
Is it noise?
It's not noise. It is a regular sinusoidal pattern. Yep. It's providing some rhythm that may be used. It's for the gait. It's the central pattern generator, the CPG, which we just talked about for the biped, right? If you cut off the head of a chicken, it will run around because it has central pattern generators in its spine that dictate walking, yeah? Or at least help with walking. They don't help with maintaining balance. For that, you need other sensory information. Yeah? So we have a total of 5 times 4. We have 20 input values at every time step. Those 20 input neurons are feeding into the hidden layer, and we're not showing the synapses because we'd end up with a pretty messy picture if we did. But every input neuron here is attached to the hidden layer back here. Our 20 hidden neurons are wired up to 3 times 12 motors in the output layer because we have 12 mechanical degrees of freedom. And the value arriving at any given motor neuron is the desired angle of that motor, the desired angle of that motorized joint at the next time step, right? Which we've seen before. So just desired angle. Current angle coming in and desired angle for the next time step arriving at the output layer. So far, so good? Okay. So we are now that's the network that's going to control the robot, but we are not evolving populations of neural controllers. We're evolving populations of CPPNs. We're going to take each CPPN in the population and allow it to paint weights onto this network. There's different ways we could do it. What are some of those ways? How would you design a CPPN not to paint pretty pictures or animations? What would that CPPN look like? What's the dimensionality of the space that we're painting into? 3D. So we'll go back to our X, Y, and Z. In the examples we've looked at so far, we were visiting particular points in the 2D or 3D space and supplying those coordinates into the CPPN, for example. So what points are we visiting in the three-dimensional space that contains the robot's neural controller? Where do we go? It's a three-dimensional space, and until we define some resolution or some locations, there's an infinite number of points we could visit inside this three-dimensional space. Where do we go?
So the touch sensors are providing some information to the
The touch, there are 4 touch sensors, that's right, and there are 12 proprioceptive sensors. So you got all these 20 input neurons. Where do we go? What are we painting in this case? We're not painting black or musical notes. What is the thing that's being painted?
For painting the weights, could the 3 dimensions be from, to, and which layer?
We're painting weights, as you said, right? So the value that's arriving, I'm just going to erase the activation function to make this a little simpler. The value that's arriving at the output layer in these CPPNs is W. It's going to dictate the weight of a synapse. But what synapse? All of them. We're going to need to paint all of the synapses here. So we need position. Position of...
Well, it would basically just be like which neuron in which layer connecting to which neuron in the other layer.
Yep, right. So if we're thinking about synapses now, we have a neuron here, and this neuron has three-dimensional coordinates. It has a particular position in X, Y, and Z space, according to this cartoon. And so does, if we're thinking about the synapse that connects, for example, this input neuron to this hidden neuron, this hidden neuron also has 3 coordinates, which gives us 6 coordinates. What do we do? Are we stuck? What's that? We can't plot 6 dimensions, so we've reached our limits. We could simplify our life by saying there is a straight line, a synapse that connects this neuron to this neuron. That synapse has a midpoint, and the midpoint of that synapse has 3 coordinates, and that's what we'll feed in, right? That's one thing we could do. So we could take each input neuron and visit each hidden neuron and query our CPPN every time we do that, and that will paint weights onto every synapse going from each input neuron to each hidden neuron. So far, so good? So we've painted half our pattern. We then go, we have the same CPPN. We go to the hidden layer, we visit each hidden neuron, each of the 20 hidden neurons, and for each hidden neuron, we visit every motor neuron, take the midpoint of the synapse that connects those 2 neurons together, take the XYZ coordinates of that synapse, and the weight that's arriving there, we paint that on to the synapse, and we have now painted weights onto our neural network. Make sense? So we've got input to hidden and hidden to motor. Are we missing anything? Let's imagine that we want to build in some recurrent connections. So we want to wire up the hidden neurons to each other. So we visit this hidden neuron, and we want to paint the weight that connects this hidden neuron to this hidden neuron. No problem. Both of these neurons have coordinates, 3D coordinates. Get a straight line, take the midpoint of that straight line, which also has 3D coordinates, pass it through the same CPPN. Now the CPPN is painting recurrent connections within the hidden layer. What about self-connections? We're going to use the same CPPN. We want it to paint synapses that go from this hidden neuron back to itself. What are the 3D coordinates we would probably want to use in this case?
We already have
The coordinates of the neuron itself, right? Okay, so good. So far, so good. So we have one CPPN that is able to paint all the synapses throughout this neural network. The sensor to hidden, hidden to neuron, the recurrent from one hidden to another, and self connections from that hidden neuron back to itself. But we have a problem. Has anybody seen what the problem is? You can imagine altering now the CPPN and it will paint different patterns of weights for this neural controller. But our CPPN is hobbled. There is a particular pattern that it cannot produce. Anybody see that yet? Let's go back to our cartoon example here. We have one CPPN that's painting the synapse that connects this hidden neuron to this hidden neuron. There is also a recurrent connection that connects this hidden neuron back to this hidden neuron, right? There are synapses that are going both ways. Tell me about the weights of those two synapses.
Same midpoint.
The same midpoint, which means they have the same midpoint, so they have those two synapses have exactly the same X, Y, and Z, which means
Same weight.
same weights. No matter what CPPN we produce, and there's an infinite number of CPPNs we could construct, none of them can paint different weights onto recurrent connections from I to J and J to I. Maybe that's a minor problem. Maybe it doesn't matter too much, but we have definitely removed one option from the evolutionary algorithm's grasp. Okay, so let's think about a different way we could ask CPPNs to paint weights.
If you add a fourth dimension, that's direction.
We could add a fourth dimension, right? So as mentioned, our intuition is now going to break down. We could add a fourth neuron called D, and we could set D to 1 when we're going from neuron I to J, and we could set D to -1 for the same set of 3 coordinates that goes from J back to I, right? So from the CPPN's point of view, it is now painting a pattern in 4-dimensional space. But we see it, the result that we see is a pattern of weights in synaptic weights in 3 dimensions. Yeah? Okay. The way that this was actually done in the paper is they just created a CPPN that had 6 input neurons, which was the X, Y, and Z of the source neuron and the X, Y, and Z of the target neuron where it arrived. Right? You could actually do it with 4. Maybe that would be a more efficient way to do things. Okay, so we got kind of a crazy evolutionary algorithm here. We have an evolutionary algorithm that's evolving neural networks. Those neural networks are painting patterns in a 6-dimensional space. We are interpreting that 6-dimensional pattern as a set of synaptic weights in 3 dimensions, which construct a neural network in 3 dimensions, which we're going to use to control a robot. We're going to watch how far this quadruped runs, as usual, or not, how far the robot travels, and the distance the robot travels. We take that one number and go all the way back to the original CPPN, and that's the fitness. We go back past the CPPN, back to the genotype that describes that CPPN, and that's the fitness we assign to that genotype. Okay, that's a lot for Thursday morning. So far, so good? Okay, so you can imagine, we better hope at this point that all of this algorithmic work is useful. We already mentioned this before. The reason we are doing this is because we hope that by biasing evolution to paint regular patterns of weights, it's going to make it easier for evolution to find regular gaits for our robots, right? Regular patterns of weights produce regular gaits. That's the hope. Yes, question?
Is there something preventing it from connecting the input layer directly to the output layer?
You could do that, no problem,
But
right?
there's not anything.
I do not think they did that in this paper. I could be wrong, but yeah, no reason why not, right? You just supply to the CPPN the coordinate of an input neuron and the coordinate of an output neuron, and off you go. Yep. Okay. So let's have a look at what actually occurred here. So they did two versions of the algorithm, the one we just described, which we're going to call HyperNeat for short form. And then in this case, they're going to do what they called fixed topology neat. So this is something sort of in between, which is they did not allow for mutation. They allowed for mutation and crossover, but not the addition and removal of neurons and synapses. So we've gone back to neat. So neat is going to evolve the neural network controllers directly, right? It's going to evolve the weights of this thing. And we're going to restrict neat. Neat is not allowed to add new neurons or synapses. It has to just evolve the weights of these. So if I back up to neat for a moment, if I back up to neat, it is going to just fix the number of synapses. And the only thing that neat can do is modify the weights of the synapses in the neural network controller. We got rid of CPPNs, everything else. Yeah? Okay. Okay. Okay, let's switch and look at a few videos before we break for today. Okay, what we are going to see in this video is one of the results from HyperNeat, and this is the worst gate they produced. They ran HyperNeat, I think about 30 times, and they ran those evolutionary algorithms 30 times, and they took the worst one from those 30 runs. And this is what they got. Not bad, right? Okay. We need the one from...
They couldn't do worse than that if they tried.
Sorry, worst is a little bit misleading here. They did evolution 30 times, and at the end of evolution, they took the best one out of the evolving population, which gives them 30 champions. And among those 30, that's the worst one. So it's the worst of the best. Thank you. Worst of the best, exactly. Okay. And I am looking for the fixed topology neat. So what you're going to watch now, this is the best of the best if you do not use HyperNeat. It's not bad. You can see how much work the evolutionary algorithm had to do in this case to find random collections of weights that produce increasingly non-random gates. Okay. Here is the same thing in figure form. We've got evolutionary time along the horizontal axis here, and they're plotting the average distance traveled by robots evolved with fixed topology neat, about 3 body lengths on average, and HyperNeat on average about 7 body lengths. So all this algorithmic effort was worth it. We are biasing evolution to search among the space of all possible weights for this thing, but to bias it towards regular patterns across the weights. One last question?
After you use fixed topology weight, neat, right?
It's just, yeah, forget the FT, it's just neat. Just evolve sets of weights. Yeah. Okay, I think we will end there today. You have a quiz due tonight, and I will see you next Tuesday. Thank you. There's no class next Tuesday, thank you. It is town hall meeting day. Okay, thank you. See you next Thursday.